This video is going to describe bacterial transformation. Remember, bacterial transformation is one of the three types of horizontal gene transfer that we are going to be interested in learning about. So generally speaking, horizontal gene transfer is when a piece of DNA is going to move from one bacterial cell, that's going to be defined by the donor cell, to a recipient cell. So that's what horizontal gene transfer is. It's just DNA, genetic material being exchanged between two bacterial cells. So no reproduction is involved, but it's just some way in which bacteria is being transferred. So during bacterial transformation, what's going to happen, and what happens all the time, is that bacterial cells do not live forever, and so they are going to die. And when a bacterial cell dies, just as when any organism dies, its DNA does remain in the environment. Now, it may break down into smaller pieces, and so this circular double-stranded piece of DNA could break down into smaller bits, as shown here. And so, as you might guess, there's going to be many pieces of bacterial DNA present within the environment. So now we look at the recipient cell. And so when there is this environmental DNA present, it may be possible for one or more of these pieces to be taken up by this bacteria. So here's a piece of DNA that was taken up by this recipient cell, and we often use the term competent to describe a bacterial cell that is able to take up DNA during this transformation process. So not all bacterial cells are equally competent. They're not all equally able to take up DNA. So this competent bacterial cell took up DNA from the environment. But remember, the DNA chromosome shown here in yellow is really the instruction manual. So right now we have this, what would be equivalent of a loose page that's not incorporated in the instruction manual yet. So through a process called recombination, that piece of DNA can actually integrate or become one with that piece of DNA that is the chromosome. And so, in summary, transformation is going to be when a dead, lysed donor bacterium, as shown over on the left, releases its DNA into its environment, and that DNA may break down into fragments, as shown here. And then there's that term, a competent recipient cell can take up DNA from the environment, but it's not just taking it up from the environment. It needs to become incorporated or recombined into the existing chromosome of that recipient cell. And so now what we have is whatever information was encoded on that new DNA fragment, this recipient cell could acquire that information and that trait.